Hey, well, thanks so much for inviting me to come here to talk. My lab doesn't really actually work in the retina, but actually today you'll see I was looking through what I've been working on. And it turned out we actually published two papers actually have something to do with retina axon regeneration. So I thought I'm going to just use that topic as sort of an example to show what I've been working on. I hope we can sort of have more discussions, but more collaborations down the road. So as you see, I changed the title a little bit. Instead of just look at RNA modification, but now I want to give two examples, look at both epigenetic mechanism and sort of RNA modification mechanism. I realized actually for the first part, actually probably echo well where's the visit today from Harvard and talk about reprogramming. So he actually involved the same pathway of DNA modification. Do this. So I just want to give a general introduction of what my lab has been doing at the Penn. So in the neuroscience department, my lab actually works on three different topics. So we're interested in neurogenesis in general. We are interested in look at the biological process, who are the stem cells, how do they generate different progenies in different brain regions throughout life. So we look at both neurogenesis in the cortex, hypothalamus, but also look at neurogenesis in the hippocampus throughout our life, look at adult neurogenesis in rodent models and in adult as well. Since I moved to Penn about 9 years ago, we also started work on glioblastoma. So we're using surgical tissues from the neurosurgery and using glioblastoma organelle as the model study how this sort of terrible disorder, what's the mechanism of others. And now there's sort of an emerging field called the cancer neuroscience, where they study the interaction of nerve system with the tumor. So many things now, many tools we've been working in the lab have become really readily sort of applicable to this field. So I've been using glioblastoma to study circuitry mechanisms, how the circuitry interact with glioblastoma, but also we're applying the glioblastoma model into clinic. So we bring that to clinical trials and look at response of organelle in the dish together with that in real patient and try to use that as a way to develop better trials for this terrible disorder. But today, I want to focus on a particular aspect of molecular mechanism control different process involving neurogenesis and glioblastoma, so which is epigenetic epitranscriptomic mechanism. So over the years, my lab has been studying the whole process of regulation from transcription, protein translation from the nucleus all the way to RNA and protein translation. So I've been working on how, for example, the 3D genome are organized, how do they interact with each other, can regulate the gene expression. And then that the sort of the chromatin histone are modified and the DNA can be methylated. So we spent a lot of time in the lab for the probably past 15 years or so and study how DNA modification can be modified in the mature neurons. In particular, we're interested in looking for how injury and neuronal activity can regulate the status of demethylation. So we've been doing a lot of whole genome bisulfite sequencing and look at the molecular pathway controls that. Particularly, we're interested in look at the reversible modification of DNA modification. So basically, look at the pathways. How does this work? That's for me to work here. Yeah.
I'll click on the bottom left icon.
So there's a point. You can see that? Yeah. Okay, great. So we're particularly focused on this demethylation process and look at how the modification can be reverted back to unmethylated C through the TET enzyme to look at this effect. So I'm going to tell you the first part story, just look at this aspect. In the second story, we're going to look at once you have RNA made, it turns out the RNA can be modified. For example, one major modification for mRNA is M6A modification, and this is through an enzyme complex which can add A onto the mRNA, a specific site, and then specific reader proteins can bind to that, and eventually can lead to downstream consequence of mRNA metabolism. So this could be RNA decay, protein translation, and others. So the second part, I'm going to tell you how this part can regulate. And the major thing looking for axon regeneration in the adult nervous system. And we start with the peripheral nervous system because we have active axon regeneration. And then once we identify mechanism, we want to test that in the central nervous system, which we use retina crushing model to look at whether the same mechanism can promote induce axon regeneration in the retina. So the first question is you look at sort of RNA DNA modification. So we're very interested in TET3, and the reason for that is because the TET3 expression. So this is in the periphery psychiatric nerve lesion model. So we cut the nerve. We look at it in DRG neurons, what happens in adult mice, and what we basically find is that if you do a lesion of the psychiatric nerve, we see a transient upregulation of TET3. So TET3 is the enzyme involved with demethylation of DNA. So it's the starting enzyme to remove the DM sort of modification. And what we find is that basically you can see after nerve injuries, there's a upregulation of expression of TET enzyme. And this leads to upregulation of the product of TET, which is TET can sort of make the MC into hydroxymethyl C. So you can see this basically again by immunostaining, you show increase of intermediate product during DNA methylation. And this is really depends on TET3, because if you knock down TET3, and now you can see the injury-induced modification is gone. So there are 3 TET enzymes, TET1, 2, and 3, and we find basically only TET3 seems to be regulated by neural injury. We know in this system, this very active regeneration. So basically, look, you crush the nerve, so this DRG nerve, if you label them, if you crush them very quickly within weeks, they can completely regenerate. So this is a part of peripheral nervous system. They have active axon regeneration in adult. Now, if you using AAV to knock down TET3, now if you do that in the DRG, you can see basically now the innervation of the axon regeneration is greatly attenuated. So that's suggesting this really required for the process. So we can look at both axon regeneration, but we can also look at innervation of the skin. So you can look at how the regenerative axon can innervate the skin at different levels. And basically, you can see basically if you block the TET3 expression, and this is greatly reduced. We can also do behavior, look at how the sensory activity of the skin are using a sort of a heat-induced withdrawal paradigm. Again, normally you see a natural recovery, and the sort of withdrawal time becomes shorter and shorter with regenerative axons, but we block the TET3, and this seems to be delayed or attenuated. So functionally, TET3 is required for axon regeneration effectively. At molecular level, we want to look at what the target genes of TET3, at least in the DRG. And we basically look at the RNA sequencing and gene expression, and we find many of genes actually the genes so-called regeneration-associated genes. So those are genes induced by injury, and they upregulate after injury, and they can, for example, ATF3 has been shown to be part of the regenerating program promoting axon regeneration in the DRG after injury. And we find not only ATG, ATF3, but many others, STAT3, TF43, others, including TET3, are induced by the injury. And that's really in a TET3 dependent fashion, because if you knock down TET3, and now you can see the induction is attenuated. And this is likely through a demethylation mechanism, because we can sequence the enhancer regions or promoter enhancer regions for the genes. And we basically find that is after injury, you see a demethylation. So the black dots are methylated Cs, the open circles are unmethylated Cs. You can see after injury, you see a decreased demethylation. And now if you block TET3, and this is attenuated. So basically, this is suggesting there's really in the DRG peripheral nervous system, there's a TET3 dependent program regulating expression, de novo expression of regeneration associated genes that involve the axon regeneration. So this is in the PNS. We also want to know what happened to the CNS. So we turn to Jigang He. So a friend, actually, we have been working together while I was a graduate student, and he was a postdoc in Mark Tessel Levin's lab. And now he's really one of the leaders in the field for axon regeneration in both in retina and in spinal cord. And his lab made an interesting discovery quite a few years ago, is although normally the DRG neurons do not regenerate after injury in adult mouse, but you can induce regeneration by knockout P10. So for example, if you get rid of P10 genes, you can see the regeneration effectively in the retina. So this is a crush model. You cut the nerve, and they can regenerate effectively. So we want to do one test in this model whether TET genes also involved and which TET genes involved. So basically, we did a screen, and by knocking down 3 different TET genes in this context to see which one is required for retinal regeneration after injury. And so basically, we knocked down TET1 to 3. So TET1, 2, and 3. It turned out in retina it's different from the DRG. It's the TET1 seems to be required for this process instead of TET3. So you can see sort of the TET3, TET2 knockdown has minimal effect, but if you knock down TET1, and the regeneration is greatly attenuated. So basically, in this model, we show that injury can change the epigenome, especially demethylation status of regeneration-associated genes, and that should be a program need to be reactivated for effective axon regeneration, both in the natural context in the PNS, but also in the context induced fashion in the CNS. I think this really leads to interesting questions. Is sort of the published nice work from Sinclair and the speaker today, and they show that if you use a reprogram approach, you actually can reprogram this process and induce effective axon regeneration in the CNS as well. So this is really about this part for active DND methylation. Now I want to turn to the next part, as I mentioned, for RNA modification. So as I mentioned, RNA really, so M6A is a major modification of mRNA. So we also want to look at, in this context of axon regeneration, whether M6A modification play a role. So we start to look at the exactly same system in the DRG, in the sciatic nerve injury. So we do an injury that will look at what happens to the M6A modification. So there's a sequence methodology allow you to look at which mRNA are methylated. So we can look at after injury, you can see the tremendous number of mRNA actually are methylated in the DRG. But if you compare that with regenerative associated genes, so those are genes that induced by injury has to be implicated to promote axon regeneration in the CNS, and you can see majority of them actually are methylated. So basically, not only they are methylated, but also these genes are induced, and you can see many of them are methylated. So you can look at the genes. GATA45 is another gene we identified to be important for active DND methylation. TET3 is for DND methylation. ATF3 is regeneration associated genes, and JUN and MYC, SOX11 are other regeneration associated genes. In addition to these genes, what's interesting is we find that many of the ribosomal proteins, so these are involved protein translation, are also show enhanced M6A modification. So we want to know what it really happened in this case. So we want to establish causal evidence. To do this is we try to knock down or knock out the enzyme mediated RNA modification. So this case, this is mediated by META3 and META14. So we knock down or knock out META14 in the DRG and see what happens. So basically here shows if you knock down META14, you see a tremendous reduction in M6A, suggesting this is a major enzyme can modify M6A. If you look at gene RNA modification gene expression levels, you actually don't see much of huge differences basically in the wild type and knockout. Basically, the regenerative associated genes are induced after injury, but the level are not changed in the mRNA level in the wild type and knockout animals. But interestingly, if you look at the protein translation, so here we do a labeling of de novo protein synthesis, we find that injury can induce a massive upregulation of native protein synthesis. People believe that this is part of the regeneration program. And then in the knockout animals, you can see this is greatly reduced. So across the proteome, basically, M6A seems to be very important for protein translation after injury. We can look at specific genes. Again, the same gene ATF3, which is induced by injury at the mRNA level, but you can see at the protein level, this is also required at the M6A pathway because after that, even the mRNA are not translated efficiently. So mRNA modification seems to be important for protein translation after injury. So functionally, we can look at again using the model. Basically, you can see the injury-induced models, and this effect is not as robust as TET3, but nonetheless, wild type and knockdown META14, you can see attenuation of axon regeneration. And this is true also look at the innervation of the skin. Basically, you see a decreased innervation of the skin if you knock out META14 for MCA modification. At the behavioral level, you also see attenuate functional recovery. In this case, we'll also look at a little bit of downstream mechanism because we think this involves protein translation. So we can also knock out one of the reader proteins involved protein translation, which is called YTHDF1. So this is a protein can bind to M6A modified transcript and lead to a protein translation. So basically, we'll find that if we knock out this gene, and then we see similar phenotype. We see a decreased protein translation, we see a decreased axon regeneration. So finally, we come back to the retina, and now the same thing. Now instead of working with Jigang he sent us to his former postdoc who actually published a paper on the P10. So now he's in Hong Kong. So we work with Kai Liu in the model. So this is P10-induced axon regeneration in the retina, and this is the case where if you knock out metaprotein, and you can see again, you see attenuation of axon regeneration. So just summarize, I told you today, basically we find the coordinated epigenetic epitranscriptomic mechanism in promoting axon regeneration. And the first phase is really involving epigenome, look at how you can regulate the expression of regenerative association genes by demethylation. And the second phase is involving how this RNA can be modified for effective translation. So all this all done by actually by a single postdoc, Ilan Wen. Now he has his own lab at Houston. Thank you.